All right, welcome everybody. Thank you for joining. We will begin in a few minutes. We're just going to give everybody a chance to log in and then we will begin. So thank you for joining. All right, maybe one more minute and then we will begin. Make sure everyone has a chance to log in. All right. Well, I think let's just go ahead and begin, and people that are continually joining can hop in a few minutes late. It's not a problem. So welcome everybody. This is a virtual tour of a few of our bioscience facilities at Berkeley Lab. My name is Kevin Gifford Tinker, and I am a protocol officer at the laboratory director at Berkeley Lab. We would love to welcome guests on site in a normal situation, but given the current pandemic, we have reimagined all of our tours to be entirely virtual. Actually, many of us on this will be working from home, but we do have some special guests who are on site, actively conducting their research. So we are excited to have them here. But this year is Berkeley Lab's 90th anniversary, and we are celebrating past achievements and imagining tomorrow's solutions. Today, in honor of our 90th anniversary, we are going to take a virtual tour of 3 of our bioscience facilities: the Joint Bioenergy Institute, the Agile Bio Foundry, and the Advanced Biofuels and Bioproducts Process Demonstration Unit, or ABPDU for short. Quickly, some few housekeeping notes. Everyone's cameras and microphones have been turned off. However, we do encourage you to communicate with us through the Q&A or chat feature at the bottom of your screen, and we will do our best to answer these questions live or save time for the end of the tour. We have enabled transcription of everything we say, so you can click on the transcription button at the bottom of your screen to view or remove closed captioning. And this tour will last approximately 1 hour, which does include time for questions and answers. So let's begin. So here's a photo of Berkeley Lab's main site up in the hills, the beautiful green hills above Berkeley, California, and Oakland, technically. But we are not going there. We are actually going to take a tour today. It's about a 20-minute drive or 5 miles down the hill to Emeryville, California, to a place we call Emery Station East. This is the gorgeous greenish building in Emery Station East. Berkeley Lab actually has the top 2 floors here for JBEI, Agile Bio Foundry, and the ABPDU. And throughout this, maybe I'll leave it back on that so you can see what this beautiful building is. You don't need to see the background that I've got right here showing. But throughout this, we will be asking you some poll questions. We'd like to keep our audience engaged and answer any questions, or maybe ask you questions too. So let's start with a quick poll question. We'd like to do this every time we have a virtual tour, but up on your screen is a poll question asking you, how far away are you from Berkeley, California? So are you less than 10 miles, 11 to 100, 101 to 1,000, 1,001 to 3,000, 3,001 to 6,000, or more than 6,000 miles away? No right or wrong answer. Just want to see where our guests coming from. All right, y'all are really quick. I love it. Okay, I think we can close this poll question right now. I think most people have responded. And all right, let's go ahead and share the results. So it looks like half of our guests are about 11 to 100 miles away. somebody or a couple of people from more than 6,000 miles away. So we thank you to those who have joined us from so far away, and to those that are our neighbors. We're glad you're here. And I will stop sharing this. I'd now like to bring up my colleague, Sarah Harmon. Sarah, I will stop sharing my screen and let you come up on screen. Sarah will be helping us as we guide you through the three biosciences facilities. Hi, Sarah.
Hi, Kevin. How are you doing today?
Good, pretty good. Enjoying the rain that we've gotten here in the Bay Area today.
Yeah, it's quite nice. I kind of always wish that it would rain when I don't have to be out in it, but it's nice because we need it.
Yeah, I agree. I agree. Yeah. So I will let you, I'll just make sure everything the screens looks good and then
Great.
let you roll with it.
All
And I'll
right.
come back on when there's some poll questions so we can launch those and see how our audience is doing. And
Sounds great. All right. Yeah, all right, here we go. Okay, so
cool.
today we're going to go over Emery Station East, and we're going to have 4 presenters. Myself, my name is Sarah Harmon, and I am the manager of outreach and communications at Emery Station East. We'll also be joined by Deepika, who is a project scientist at JBEI, Emily, who does public affairs for ESE, and Laura Fernandez, who is a senior research associate at the ABPDU. So next slide, please. All right, so this is our gorgeous building. As Kevin mentioned, we are not in Berkeley, we are in Emeryville. And here at Emery Station East, our programs are actually associated with a lot of different organizations, including Lawrence Berkeley Lab, but we also are affiliated with other national labs. And we occupy the 3rd and 4th floors, and we have about 230 employees for all of the programs occupying these floors. So we're gonna go on to the next slide. Great. And so we will start our tour today at the Joint Bioenergy Institute, but I wanted to point out in this diagram here what's so important about the work that we do together. Sometimes people get a little bit confused over what the difference is that we do. So at the Joint Bioenergy Institute, we do basic research. We do really fundamental research and really figuring out, in our case, how biology works and how organisms work. And then our colleagues at the Agile Bio Foundry, they work on system testing. And what they're really trying to do is kind of do a proof of concept. So they're working on, we at JBEI will find something that maybe we'll find a new enzyme, and then somebody else will have an idea on, oh, we should use this enzyme to do X. And at Agile Bio Foundry, they can prove that concept, and then they would move on to the ABPDU, the Advanced Biofuels and Bioproducts Process Development Unit, to scale that up to see, okay, so we tested it out, it works. Now, can we actually make that available? So we're going to learn a lot more about that in a little while. And I do want to point out that we use a lot of acronyms at the lab. So if one of the ones we use confuses you, please let us know so we can clear it up. And the same goes to any other terms that we might use to talk about our science. We recognize that we use these words all the time, so they are familiar to us, but they might not be very familiar to you. So let's jump right in and get talking about JBEI. Great. So as I mentioned, while we are physically, our headquarters is in Emeryville, California, and our physical headquarters is associated with Lawrence Berkeley National Lab. We actually have about 250 folks all across these JBEI sites. So we have folks in Washington, we have a lot of folks in California, We have folks in New Mexico, New York, even Illinois, Iowa. So we have people all over the country who are working to find biofuels and bioproducts that are going to be one of the next energy solutions for our country. Next slide, please. And on top of that, we are one of 4 bioenergy research centers. So all of these research centers are funded by the Department of Energy. And as you can see, they are similar to JBEI in that they are headquartered in one location, but they have partners all across the country. And in fact, we have laboratories at Lawrence Berkeley National Lab that are partners with more than one of these bioenergy research centers. So the others are CBI, or the Center for Bioenergy Innovation, which is led by Oak Ridge National Lab in Tennessee. We also have GLBRC, or the Great Lakes Bioenergy Research Center, which is led by the University of Wisconsin in Madison. And we also have the Center for Advanced Bioenergy and Bioproducts Innovation, or CABI, which is led by the University of Illinois at Urbana-Champaign. And so each of these BRCs has some very specific individual goals, but because we're all charged with resolving the same basic problem, we also frequently collaborate and share the information that we find so that we can get the job done together. So now we are going to head into JBEI. And so what you're seeing here is a hallway on the 4th floor, and this is a very long hallway, and it is lined with green windows. And so we actually affectionately refer to this as the Green Mile. And we'd like to note that the majority of the photos and visuals that we've used today in our tour were actually taken before the pandemic. And so that is why you won't see people in masks, but you will see some people in masks later because they're actually on site right now. So essential members of our team are working on site, and we have reduced capacity and are taking all required precautions to keep our team safe. And so now we're going to talk about the questions we're going to answer today. So we're going to talk about, what does JBEI do? What are biofuels and bioproducts? And some of the discoveries that have come out of JBEI. And now, next slide, please. And we have a poll just to get us thinking. So which of these items are usually made from fossil fuels? We've got lipstick, toothpaste, crayons, aspirin, and lotion.
All right, so I will launch this poll question. This poll question is unique from the past one, that this is multiple choice. So we'd like to see what you all think is made from fossil fuels. So on your screen, you should be able to select, do you think lipstick, toothpaste, crayons, aspirin, and or lotion are made from fossil fuels? Please go ahead and vote, and then we'll see what happens. Some good feedback already. I like seeing how these come in. But I'm going to guess something after we end this poll.
Yeah.
For Sarah to mention, but let's see. All right, I think most people have had a chance to vote, and let's end this poll. Thank you so much, everyone, for voting. And I will share these results, but let me just make a guess. We'll see. Crayons. Okay. Most people said crayons are made from fossil fuels. Lipstick also, which is interesting. Lotion next, and then toothpaste. And no one said aspirin, but Sarah, do you want to give us a little spoiler alert, or spoil it now?
Yeah, sure. So this was a trick question. All of these have components made from fossil fuels. Every single one of these items.
Wow.
The reason that we do what we do at JBEI and the other ESC facilities is because we know that there is a limited quantity of fossil fuels. And so if all of these things that we're using all the time on a daily basis, multiple times a day for some of these, that we know that if we're using these, that we will eventually run out. And so that's why some of the work that we do is so important. And I think I was sharing the other day when we were talking about this poll that when I was a kid, my mom would always tell me, oh, when you go to bed so your lips don't get dry overnight, that you should put Vaseline on your lips. And so I've done that my whole life. And it wasn't until like working at JBEI that I kind of started looking at the container and I realized Vaseline's actually called petroleum jelly. Like I should have known this whole time that I'm basically smearing fossil fuels on my lips at night. So now we're going to get into the work that we do at JBEI and what we're trying to do to get better ingredients for some of these products. So the mission at JBEI is to develop basic science and advanced technologies to enable the conversion of plants. So you see here, there's a plant on the left-hand side of your screen. So what we're really doing here is going from plants, going through a lot of chemical and biological processes to be able to get it into a version that we can use to make biofuels and bioproducts. And a major component of our research is to make sure that these technologies are commercially and economically viable so that they can be adopted into the real world. So not only do we do this research, but we actually make this technology available to other organizations and industry so that they can use it too. And one very, very important thing to mention is that the Department of Energy actually funds us about $25 million a year to do this research. And next slide, please. So at JBEI, we have 5 divisions. And so we're going to kind of take a look at these divisions, but also we're going to just give a brief overview of how these divisions relate to the process of making biofuels and bioproducts. So our first division is feedstocks. And what the feedstocks really are is what you can see here is it's the plants that we start with. We also call this biomass. And then we have the deconstruction process and the deconstruction division, which is really focused on breaking these plants down. And then we have the biofuels and bioproducts division. And what they're focused on is making the highest quality ingredients at the best price to be components or products to replace those that we have that we currently use that are made from fossil fuels. And then we also have these 2 other divisions, the technology division and the life cycle and economics division. And both of these divisions are really important because they are a part of the entire pipeline. The work that we do, it informs the technology and life cycle and economics divisions, and the life cycle and economics and technology divisions help us make the decisions about the work that we're going to do and the directions we're going to take in our research. So, and as you can probably tell, the work that we do is really cross-functional and it's multidisciplinary. So at JBEI, we have engineers, software developers, physicists, plant biologists, simulation experts, and way more specialties than I can even mention. And they're all working together side by side. And we really promote this collaboration and sharing of research because we believe that people from different perspectives are going to be able to help us get the best answers. And so now I would like to introduce you to my colleague, Deepika, who is a scientist at JBEI, and she's going to tell you more about all of the research we're doing and tell you more in depth about our divisions.
Thank you, Sarah, and welcome everyone. Thanks for joining us today. Emily, next slide, please. Yes. So as Sarah was mentioning that to achieve JBEI's mission, we have these 5 divisions. I will be going and talking about for a few minutes and giving you a brief on each of these divisions to give you an insight of what is actually done in those and how that enables the overall JBEI goal. So each of these divisions have an individual milestone, but the end goal is the same. So they are also very collaborative. In the feedstock division, so let's start with that. The life on our planet Earth is carbon based, and every thing that you see running around is running on energy that they consume from carbon. That carbon, very fascinatingly, is carbon dioxide, which is fixed by plants by a process called photosynthesis that you may all have heard about. So plants use this process of photosynthesis to fix CO2 into glucose and fructose, the very common sugars that we hear about. But then there are so many other type of sugars and molecules that plants make because they grow on those sugars, and they make their body and everything derived from those component sugars, which could be xylose, arabinose, mannose, and phenolic compounds that make lignin. So a plant body is made up of these 3 polymers: cellulose, hemicellulose, and lignin. Cellulose is the polymer which is actually made of repeating units of glucose. And hemicellulose is a polymer that is made up of repeating units of xylose. And lignin is a very complex and recalcitrant polymer, which scientists are still figuring out. What we do in feedstock is we try to engineer non-food crops that can grow faster, make more cellulose, because cellulose is the most easiest polymer to access because it's made of glucose. And also they should be recalcitrant so that they can withstand the harsh environment that they grow, and they use less fertilizers, and they can be easily processed down in the later processes for extracting sugars out of those plants. So that's the research we do in feedstock division. And here is a nice schematic figure where you see a bioenergy crop, individual plant cells, how that plant cell wall is made of. There is a yellowish lignin cellulose fiber, which is wrapped around by hemicellulose fibers, and that is strongly wrapped around by lignin fibers. So next slide, please. So after doing the benchtop research on constructing these plants with the traits that researchers like in a bioenergy crop, the next step is to take them into greenhouse. So if this was an in-person tour at JBEI, I would have taken you to these greenhouse plant growth chambers and shown you around. These plant growth chambers have controlled humidity, temperature, and features that enable us to feed them with the controlled fertilizers and things we want them to grow at and test them at. So after, and one important thing is that we don't use any pesticides and insecticides. Instead, we use these carnivorous or you can say insecticide or pest trapping small plants. The pictures that you can see in the at the bottom. Sorry, Emily, can you have the laser pointer on? Yeah. So can you like scroll and show as I'm talking? So you see these small plants at the bottom. These are the insect trapping and pest trapping plants. So we don't use any insecticides and pesticides, chemicals in this soil. After the plant growth chambers, we move these plants, the ones which have the traits and characteristics we would like to further study, into actual fields. Next slide, please. Next slide. Sorry. Yes. So what you see here, these are not trash bags. These are actually the energy crops that were grown, and they are properly bagged to then taken back to our JBEI for analysis of what we predict as sugar content, lignin content, and are they easily accessible and processed, can they be processed down. So the scientists you see in the field, they are working on tagging these plants properly and bagging them and bringing them to JBEI. So after that, next slide. So that's basically the research at Feedstock Division to make plants more accessible to the downstream processes that will be done in the Deconstruction Division. So we'll move to the Deconstruction Division now. Now, research in Deconstruction Division, to give you a perspective, is what we do at Deconstruction is like, so plants use CO2 and photosynthesis to make up their body. And that is, the plant body is so recalcitrant to the harsh environment around that you see them growing in like sub-zero temperatures and they can still stand. They live for 100 and 200 years and there is no biological attack on them that they are going to, they fall down because something happened. So the plant body is quite recalcitrant to these things. We cannot just break down them with just easy available chemicals and biological processes. So it's a very tough thing to find, to be able to extract these sugars, but we have been successful in getting almost 90% sugars out of these woody biomass. So what you see here is that we have two processes, an integration of two processes, a chemical process and a biological process through which we take out the plant sugars. So Emily, can you go through the bottles? So we start from the biomass, which is the very first bottle on the top, which you see is a woody biomass. Sorry, sorry, yes. So the biomass is the first bottle, and we chip down this biomass into powder form and mix them with these ionic liquids. Ionic liquids technology is very unique to JBEI, and ionic liquids are salts which remain as molten liquid state in room temperature. And because of this, they have very unique chemical properties. And once the plant biomass is mixed with ionic liquids and is heated up, we get this third bottle, which is a very dark slurry. And this biomass and lignin mixture can then be processed or separated. And what we get is the fourth bottle, which is actually cellulose. And lignin and everything goes away with ionic liquids. These sugars, or glucose, which is trapped in cellulose, can then be released by using biological enzymes called cellulases that will cleave the cellulose into individual glucose units. Then we move to microbes, which will ferment them and make them biofuels, which is the last bottle. So all these things are for in-person display if you happen to come to JBEI once the COVID pandemic goes. Next slide, please. So as I mentioned earlier, the research at JBEI is at Deconstruction Division is a combination of both chemical and biological processes. So the ionic liquid work was in the previous slide. Here, I also want to talk about that we also work on these enzymes that are going to break down cellulases to glucose. That would also break down xylose, which makes hemicellulases, and utilize that also as a carbon source for products. Next slide, please. So as we have talked about the Deconstruction Division's goal, a brief overview on that, we'll now move to the Biofuels and Bioproducts Division, sorry. So research in Biofuels and Bioproducts Division is to engineer microbes that will take up these sugars that we have released from the plant biomass after deconstruction and make them into fuels or other commodity chemicals. Now, the process that we utilize is basically fermentation, as you may have heard about, like, wineries and making vinegar or sauerkraut. So a same microbe can make alcohol by fermenting sugars, and then there is another microbe that can make an acid by fermenting those sugars. So at the Biofuel and Bioproducts Division, we have understood that process, what makes acid, what makes alcohol, and we are trying to explore it further to make the molecules that interest us. And the mission of JBEI, that is making fuels and other commodity chemicals. So at Biofuels and Bioproducts Division, we make tools, develop, we work on different microorganisms and explore their pathways of, how we can exploit them. Please, next slide. So at the end goal of this BBD division is to make drop-in biofuels. So drop-in biofuels are is a simple term used for fuels that can be used in our existing automobile industry, from jet fuels, truck fuels, and car fuels. We don't have to change our existing automobile machineries for another alternate technology. And you can just go to a gas station and fill your car with the same fuel, with a biofuel instead of a fossil fuel, and that will be a drop-in biofuel. So we are trying to make molecules which are structurally and in energy value are very close to gasoline. Next slide, please. So with the fuels, there is also an interest into making other commodity chemicals. If you remember from the poll that you just took a few moments ago, and you must be surprised with the kind of products we can make through fossil fuels. So when the crude oil is extracted, it's not just only processed into gasoline, but all the other side products which come out have found their use in lipsticks and paints, and everything that you can see around your room today will have some or the other form of chemical that was derived from those fossil fuels. So if you're making fuels, biofuels as a replacement, we also have to find alternative for all those other chemicals too. So the bioproducts defined all those other chemicals that we are exploring to make. And here is one very successful work that was done at JBEI. It was making an indigo dye called indigoidine. And you can see in this reactor or vessel, there is a dark blue dye. So this is a microbe which produces dark blue dye from sugars. So instead of making indigo dye from harsh chemicals, now we can make it sustainably and with biology. Next slide, please. So this is about the feedstock, plants, deconstruction, chemistry and biology, and microbiology and synthetic biology tools. Now we move to the technology and the life cycle analysis divisions. Next slide, please. So this is our lead division, you can say, life cycle economics and agronomy. And this is a very important division because though our goal at JBEI is dedicated to basic science, but our mission is also to make it commercially available, to make it more economical. So at every step of the work or technology that we develop, whether it is feedstock, deconstruction, or bioproducts division, it is very important to see how economical that is in a commercial point of view. And the life cycle economics division helps us in identifying that which direction we should pursue this our research, or what will be a very costly step, and we need to find alternative for those steps. So that's the work which is done at lead division. Next slide, please. So now we move to the technology division. Next slide. The research and technology division is very enabling for all the researchers. So imagine that as a human being, one can do like say 100 samples and study 100 samples in like 4 hours or 5 hours. But with the use of robotics and instruments, one can do 500 samples in those 5 hours. So that is the way this technology that we have at JBEI is enabling the scientists to speed up the process and research that we have. We can also work in quantities of chemicals in microliters that are very hard to pipette manually, but with the use of robotics and instruments we can do that. And so in the robotics and technology division, we are the researchers who work there, they are trying to enable the bench scientists in speeding up their research, in enabling them to look for more products, more samples, more substrates, and more reactions. Next slide, please. So as Sarah was mentioning earlier, that the work at JBEI has been very collaborative with not just between the divisions, but the people who work here come from very diverse background. And with that diversity, we have been diversity of thoughts and science, and these divisions, we have been able to reduce the cost of biofuels from about $500,000 that was 14 years ago to around $37. And with the use of the side products that come out from the biomass pretreatment, finding use for them, we can further lower the cost of biofuels to around $2.50 per gallon. Next slide, please. We hope you enjoyed seeing a bit of JBEI today and learning about the biology that we do. And we are trying to integrate this biology to the real world and the solutions that we are finding. And now moving forward, we will learn about the other divisions that we have at ESE. And I would like to introduce our next speaker, Emily. She will be talking about the Agile Bio Foundries.
Okay, hello everybody. So now we are going to go into our next program that is represented at Emery Station East, and that is the Agile Bio Foundry. So with the Agile Bio Foundry, we move a little bit down the pipeline of technology advancement, as you can see in this graphic that we're revisiting here. So the mission of the Agile Bio Foundry is to accelerate biomanufacturing and build the US bioeconomy. So the Agile Bio Foundry is actually a collaboration between 7 different national laboratories across the country. So we have researchers at Emery Station East on the 4th floor, and then we have researchers all across the country at other labs. So our work focuses on biomanufacturing. So that is using engineered microbes to create bio-based chemicals and materials, otherwise known as bioproducts. So this is what our work revolves around. So if you think about our name being the Agile Bio Foundry, if you think about what a traditional foundry is, what you think of it as, a place where they use metal and mold metal into different shapes in order to achieve different functions. So a biofoundry is pretty similar, except the material that we work with is microorganisms. So we are molding, we are molding microorganisms, engineering them to achieve specific functions and produce various chemicals and compounds. So one of the biggest problems when you're developing a bioproduct, is just how long it takes. So it takes 10 years on average to develop a bioproduct. So that's a pretty long time. So the mission of the Agile Bio Foundry is to cut this time in half. And the way that we're doing this is by developing new tools and technologies that help make this process more efficient. So the way that we do this is through our collaborations with industry and academia. So these are industry partners and academic partners, researchers who are in this biomanufacturing space. So our collaborative projects really focus on solving common problems in bioproduct development that a lot of people face. So through these partnerships, we're developing new methods and looking at things like how can we save time, how can we lower the cost, how can we boost production, make things more efficient in the biomanufacturing process. So why do researchers come to the Agile Bio Foundry? Why would they want to come to us to work with us for this purpose? So we like to describe the Agile Bio Foundry through this analogy of public transit. So say you need to get somewhere, you're going to a restaurant or something, you might get there by the subway. So you would take this public transit that's publicly available to you, but it might not get you all the way to your destination, right to the door of where you're trying to go. So in that case, you need to take a walk, take a bike, take an Uber or a Lyft or something like that in order to actually get to your final destination. So the Agile Bio Foundry sees itself in a very similar way. So our partners come to us wanting to get to a specific destination. And in this case, they're wanting to make a specific bioproduct. So if they work with the Agile Bio Foundry, they don't need to reinvent the wheel. So they have this publicly available resources available to them through the Agile Bio Foundry. We have state-of-the-art technology, capabilities, and expertise that's otherwise really hard to find. And so through the Agile Bio Foundry, we can help them get pretty close to where they need to go, pretty close to producing the bioproduct of their choice. And then after working with us, our partners can take the knowledge they gained through our collaboration and make it the last mile to their destination and successfully making the bioproduct of their choice. So now we will head to our next stop, which is on the 3rd floor of ESE, the Advanced Biofuels and Bioproducts Process Development Unit. And as you can see here, the ABPDU is dealing with the furthest down the pipeline of technology advancement and really working on commercializing biofuels and bioproducts. So for that, I will introduce Laura, who is a Senior Research Associate at ABPDU. And she's actually on site today, so I'm glad she could join us, but hand it over to you, Laura.
Thanks. Yeah, so next slide. Welcome to the 3rd floor of ESE. This is the ABPDU. I'll spare you the long name this time. So what you're looking at is about half of our fermentation room. I'm a fermentation engineer at the ABPDU. So the ABPDU is a collaborative facility that partners with companies and academic researchers developing biofuels and bioproducts. The ABPDU acts as a bridge between science and industry by helping companies develop, improve, and scale up their technologies so they can be commercially viable, which is a really important step in taking things developed at places like JBEI and through the ABF and putting them in the real world. At ABPDU, we scale up science, so we can take something from lab scale to production scale. So researchers engineer microbes to produce a variety of valuable compounds in small quantities at laboratory conditions, kind of like the plate you see on the left-hand side of this slide. But scaling up isn't as simple as just multiplying the recipe to take it up to industrial levels. And you kind of face new challenges at each level of that scale up. One of the challenges in that is that very few facilities are able to sort of see this process from small to large scale, whether that's because they have limited space or not enough money to invest in their own large scale equipment. So the ABPDU is one of the rare facilities that can help researchers and small companies take their technology from small to large scale. So industry and academic collaborators come to the ABPDU to prove their technology can work in the real world. ABPDU is able to take technologies from the very small scale. So we're talking about something developed at like the size of a glass of water. And our largest piece of equipment is like the size of a standard bathtub, which is normally enough, kind of the goal mile marker that the ABPDU helps companies get to is like a first kilogram of product. Which really helps in proving to society and investors that it would be viable to scale up the technology further. So next slide. I think Kevin will come back on here.
Yes, sorry. Hi, how's it going? I took my headphone out. But yes, we have a poll. So this is a choose your own adventure that we have for you all to select. And I will reiterate, we're getting great questions in. Sorry, that's why I was a little distracted right there. But let me launch this question. What product would you like to learn more about? So we will launch this. This should be able to select one.
Yeah.
Would you like to learn more about indigo dye made from microbes, skis made from algae oil, animal-free collagen, a key component of infant formula, or municipal waste? So these are
I should mention, yeah, all of these products have been done at the ABPDU. So just for the sake of time, pick your own adventure.
Cool. And I think we'll do one, and we'll see how the answers turn out after everyone's had a chance to vote. I think we've got one winner, and then we will, after that, go into some Q&A. So looks like most people have voted, and a little over a third would like to know about animal-free collagen.
Interesting.
I'm curious about that.
Yeah. Yeah. I'm gonna close this poll. Yeah. So this is a company we worked with called Gelator. And so collagen supplements are pretty popular nowadays, but these supplements often aren't vegan. So a biotech company located in the Bay Area is developing a bio-designed animal-free collagen. But they didn't have the laboratory space in which they could attempt to do any of their scale-up technology. So the ABPDU was kind of an essential partner in that. In a few years' time, Gelator was able to work with ABPDU to demonstrate proof of concept and commercialize their technology. In 3 years, their technology was scaled up from a test tube to a 30,000 liter scale, which is like 3 years is nothing. Kind of as Emily mentioned, it typically takes up to 10 years. So it was really cool to see a company go from like a baby idea all the way up to a commercially viable product. And that's the two collaborators at our 300 liter tank, which is like our bathtub tank for sizing reference. Yeah, and I think that's all I have for you today.
All right. Thank you so much. I think that wraps it up, but we will now go on to some Q&A. If I could invite my colleagues back up on screen and we can kind of go through some of the questions that we received. And I think the first one that we got in was, so as a question, this is actually a really good question. With cars going electric, why does JBEI and the facilities at Emery Station East, why are you still working on fuels? That's an interesting question.
Okay.
Someone want to answer? Okay.
Yeah, so that's a good question. It's important to look for all the aspects of alternate form of energy that we have around us, but look at the current automobile industry. We have like, I don't know, $1 trillion invested into the current cars and infrastructure, and not everyone has the money and finances to go out and buy an EV vehicle. Like, for example, all the EV vehicles right now, even the smallest one cost above $30,000 or something. And Tesla is very expensive. So going to biofuels, one can use their own cars. They don't have to find car replacements and just use these fuels. So that's the reason.
Okay. And the fuels are also not just for cars, right?
Yeah, they're not just for cars, for trucks, for planes. Yeah.
Awesome. And we had a question that came in about artificial chloroplast. Do you all have any knowledge of that at Berkeley Lab?
I don't think we are working on artificial chloroplast. In my understanding, artificial chloroplast would be introduced into an organism that naturally cannot fix CO2. So because we directly work on plants, we are looking into improving their native pathways, you know, how they can fix more CO2 or how they make less lignin and produce more cellulose. So trying to alter their natural pathways instead of expressing an artificial chloroplast in them. And that's my understanding, yeah.
Another interesting question. What can you make plastics from the biofuels you're making?
Yeah, actually my PhD work was on plastics.
Oh, really?
Yeah. So since plant feedstock, you know, once you have sugars, as I was explaining, one can make alcohol, one can make acid, right? So you can make fuel and you can make other chemicals too. So there are like chemicals like lactic acid, succinic acid, malic acid, or polyhydroxyalkanoates that can be made from these microbes by fermenting sugars, which are actually a substitute for bioplastics, or petroleum plastics substitute, and they will make bioplastics. Yes.
Yeah, and like the same way that you kind of pull oil out of the ground and you don't look at that and see plastic, you can take oils made from biological material or organisms and process them sort of in a similar way and refine them the same way you would oil to get it into forms. So actually like the algae oil ski project is a good example of that. Where they took what we gave to the collaborators was oil, and then they had a process that they refined that into plastic that could be made into skis. So yeah, it kind of works both ways. You can either make the plastic itself out of organisms or refine a bioproduct into plastic.
As a snowboarder myself, I would love to try out those a snowboard made out of
yeah, I think they launched in
bioproducts.
2019. They actually had a working product. So hopefully in the next couple of years, we'll see it more commonly.
Cool. I'm down to try it. That sounds like a lot of fun. Actually, going along with that, Laura, what kind of plants do you work with? And Deepika or anybody can answer this question. What kind of plants or microbes do you work with?
On the plant side, they try and focus, which I think Deepika mentioned, on non-food biomass and often like woody biomass, so that it's not taking the place of like space that we could be using for food crops. And then in terms of organisms, I do some of the biosafety work here, and we see just about everything. But a lot of like E. coli and yeast-based products, those kind of form up the general bulk of what we do.
And being specific about plants, I think there are two categories. One is grassy and one is hardwood. For the grassy, I think it's sorghum and switchgrass that we work at JBEI. Then there are another energy centers which work on hardwood processing like eucalyptus and poplar plants. Since Laura is in ABPDU, that is very much into like once you have a success, that's when you go to ABPDU, right? You can make things lab scale. But at JBEI, because we do basic science, so we also work on organisms, which are like pseudomonads and bacilli and corynebacterium. And so we are developing technology on that. And once they are successful, I'm sure Laura will be working on those, scaling up those processes. Yeah.
Fingers crossed for sure. And then I guess the last question we received, and then we will maybe wrap it up. But what kind of major? We have some people that are asking about contact and wanting to get involved. What kind of majors do you recommend somebody to have? You mentioned, Deepika, you worked with plastics as your part of your major, but what would it be a major for someone to have in their college or post-secondary school?
So I'm a chemical engineer by training, and I think kind of most of the engineering team here is some variation of chemical or biochemical engineers that are working on kind of the reactor work. But then we also have just kind of a plethora of science majors doing analytical work. So there are a couple different ways to get involved from an ABPDU perspective.
Cool.
I have an undergrad in engineering in biotechnology, and I came to do a PhD in microbiology in the US. And then I joined Berkeley Lab as a postdoc, and now I'm a project scientist. So, but around me, I see my colleagues who have come from a very like strong physics background or chemistry background, who are researchers here, and software engineers. And I mean, Sarah and Emily also work here. I don't like, right? So that is like, it depends upon what kind of work you want to do at JBEI. As we were showing, there are like different technology divisions. Not everybody is a scientist. People are also here with undergrad degrees in economics, and I think communications and writing, I think. And yeah, so it depends upon what sector of JBEI you want to contribute in. Yeah.
All right. Sarah, Emily, you want to any last thing to reach out? Or I think Deepika maybe says it perfectly. You can find careers at JBEI, ABPDU, Agile Bio Foundry, and Berkeley Lab in general with a science background or with other ones as well. Doesn't necessarily need to be. I don't have a science background, so I'm here as a layperson trying to figure out what's going on.
Yeah, I always like to say that I have intentionally led a science adjacent life, but I am not a scientist. And that's okay, because we need people like us that really care about what's happening in science, but are able to communicate that to others. And I also just wanted to point out about the career opportunities that because we have we did explain kind of these unusual structures that our organizations have. And so we do, of course, have positions open on the LBL website, but sometimes positions from our partners are located at our Emeryville facility, but you have to apply through another organization to work there. So we highlight those on our social media. So please follow us if you're interested, because that's the best way to find out about those, because most people aren't going to go look at Sandia National Labs to work in Emeryville, but in fact, they do sometimes have positions there. And I'm also going to put a link in the chat right now for some opportunities we have for high school students and undergraduate students, because we do take very seriously being able to grow up the next generation of scientists. So I want to make sure that people know about those opportunities.
Awesome. Thank you so much, Sarah. So I'd like to thank Laura, Sarah, Emily, and Deepika for your help and showing us what's happening at Emery Station East, at JBEI, Agile Bio Foundry, and ABPDU. I hope you all remembered those acronyms because we mentioned them. But as I mentioned, and as you can see from all of our backgrounds, this is Berkeley Lab's 90th anniversary, and we encourage you to visit berkeleylabnext90.lbl.gov. You could see it if the Zoom background works. It's right up here at the top above all of our heads. So berkeleylabnext90.lbl.gov. And as we celebrate our past and we imagine our future, we can join us for other events like a speaker series, podcast, and yes, other virtual tours. So we have a general tour and where you can learn about Berkeley Lab's history and some other of our facilities, and other deep dives of other of our biosciences and other areas of Berkeley Lab that you may not knew existed. So we hope you sign up. You can see those at the website linked above, and we'll send a follow-up email with some of these links that we spoke about today, so you can learn more and reach out to us with any other questions you may have. So I want to thank you all for joining again, and I hope you have a great day. Enjoy the rain if it's still going on here in the Bay Area. But all right.
Thank you, everyone.
Bye-bye.
Thanks.